This video abstract is AI generated. How does TRPV3 convert heat into opening of an ion channel gate? TRPV3 is a tetrameric cation channel involved in temperature sensation. It sits in the membrane and contains several functional regions, including the voltage-sensing-like domain, the pore domain, the TRP helix, the helix-stern helix, the interprotomer coupling domain, and the ankyrin repeat domain. Cryo-EM structures have shown closed, sensitized, and open states of TRPV3, but these structures are snapshots. They do not explain which residue interactions sense temperature, how that information is transmitted through the channel, or how distant regions control opening of the lower gate. To address this, we used Hamiltonian replica exchange molecular dynamics to sample TRPV3 across 28 effective temperatures. For each temperature, we calculated residue contact frequencies. Principal component analysis then separated these contacts into temperature-sensitive modes. The first mode captures the dominant melting response. Many contacts decrease in frequency as temperature rises. Other modes show more subtle, nonlinear behavior near the activation and sensitization range. This means that temperature sensing is distributed across multiple contact modes rather than controlled by a single switch. Mapping these modes onto the structure revealed that they cluster in functional regions of TRPV3. Some contacts localize to the ankyrin repeat domain, where large bending motions dominate. Others cluster near the helix-stern helix, the TRP helix, the voltage-sensing-like domain, the interprotomer coupling domain, and the S6-TRP bend near the lower gate. Network analysis then showed how these distant regions communicate. High centrality contact pathways connect the ankyrin repeat domain and interprotomer coupling region to the helix-stern helix, TRP helix, pore domain, and lower gate. In this model, thermal information is transmitted through an allosteric contact network. Finally, random forest analysis linked contact states to gate expansion. Specific temperature-sensitive contacts were predictive of whether the lower gate was closed or expanded. Importantly, the predictive contacts were distributed across the channel, supporting a model in which gating is controlled by several coordinated dynamic modes. Together, these results show that TRPV3 uses distributed temperature-sensitive contact modes to convert heat into pore opening. Temperature changes reshape residue interactions, reorganize allosteric networks, and regulate expansion of the lower gate. To know more, read our work online.